Hi, I'm Peter Cook. Welcome to the pan-genomic model, part 6. In the first 5 sections, I provided a framework with which to understand structure and function. In this section, I want to give you a simple formula that enables you to predict the activity of your favorite gene, something that looks like this. Now, there's a long and continuing history of attempts, and everything I've told you tells us it must be foolish to try to derive such a formula. For example, when thousands of regulatory loci around the genome affect the activity of any particular promoter. Particle physicists, in the early days, when they were smashing atoms apart, faced an analogous and intractable problem, which meant that they were making little progress. But then along came Feynman, and he said, focus just on the interactions that dominate the outcomes. Unfortunately, I collaborate with a physicist, Davide, who understands this history. So let's focus on this small segment of a genome and ask the question, what dominates how often R will fire? I think there are 2 critical factors. They're both affecting the first steps in the pathway. One is obviously going to be how often R will bind to a factory. And the second will depend upon how far from that factory R is. So let's say S is already being transcribed, R will be tethered very close to the factory, and that makes it the likeliest loop. And so it's going to be that distance in 3D space between R and the lower factory that will determine how often R will fire. 2 structures determine how often R will fire. First, the simplest possible model, where all promoters have the same affinity for a factory, and factories are only of just one color. However, we know that genic promoters tend to be more active than non-genic ones from the GWAS data. Active promoters have different relative activities depending upon which type they are. Genic ones tend to have the highest activity, enhancers in between, and there are some promoters that are nevertheless active which are on neither of those 2 lists, and we're going to call those other. So we go to the GWAS data and we say the activity of the other group is 1. Enhancers are going to be 3 times more active. The sole promoter of the 5 at the top that is a gene is going to fire the most often. So now, because Q is often firing, R will find that it is often tethered closer to a factory at the bottom where Q is active than to the factory in the middle in which S was active. So it's now going to be these 2 Feynman diagrams. Here is the formula. It couldn't be simpler. The 2 terms in the brackets are going to apply to our 2 Feynman diagrams. B is a constant that captures concentrations and promoters, numbers, and so on. C is a related constant that captures the same kinds of concentrations and affinities, but in addition captures 3D distance effects. The sole variable, the number of base pairs to the next nearest active promoter. Here is the obvious question. So we look at one particular region of one chromosome in this cell line, And we go to all of the active promoters, which we can position using, for example, DHS sites. So once we've located the active promoters, we go and apply the formula to each one in turn and arrange the output in rank order from the most active promoter to the least active one. So if we go to this particular gene, it turns out that it's quite active in these cells. It's close to the top of the rank order. This enhancer is below it, but still pretty active. And so we go down the chromosome, applying the formula to each one, and we get the answer from our computer in no time at all. And then we're going to compare our results with data obtained using the method that is the standard choice, RNA-seq data, which of course lacks information on most enhancers. But the really good comparison is with GWAS data, so that's going to be our gold standard reference. So we go to the whole chromosome and compare results obtained with our formula to those against GWAS and poly-RNA-seq. And we work out the Spearman correlation. So this is a really, really stringent effect. And if there's a perfect match between our formula and the GWAS data, then we will get a correlation of 1. That means we've got every single promoter in the cell in the correct rank. We do very, very well, and I think you can see that we do even better in seconds at essentially no cost than RNA-seq. So now I'm giving you results for every human chromosome in HuVex for the 1 and the 3 state formula. And now I'm comparing it with the RNA-seq data. And you can see that the formula is outperforming RNA-seq for all but 2 of the chromosomes. But of course, the formula is providing a lot more information on all of those enhancers. We want perfection. How do you think we could achieve it? Well, the obvious way is to add more Feynman diagrams. And we have only thus far considered cis contacts. Now it turns out that trans contacts outnumber cis contacts in all methods other than Hi-C. And if you want to see the data that backs up that statement, you have to go to my section 8, the frequently asked questions. And we have only just one kind of transcription factor, one kind of factory, so we need to add more colors. In other words, we need to define all of those small world networks. Now recall that I've used the ratios in the 3-state formula that were derived from GWAS data from HuVex. And it turns out that those ratios can be applied to other cell types to give roughly equivalent results. So the ratios, in other words, are transferable. So to summarize, I told you we had an intractable problem that we've managed to simplify using Feynman diagrams, and we're now in a position to very quickly shuffle and manipulate results in seconds on our computer to find outcomes that might be of interest. So we just have 2 critical inputs in the 3-state formula. We need to know where all of the promoters are, which we can get from DHS sites or GWAS data or histone marks. I've provided you with some transferable constants, and if you want to validate results, you need some GWAS data or equivalent. The formula is derived from first principles. Importantly, and unlike other models, it's fitting free and it doesn't use any contact data. How can we improve it? I'm hoping that we can derive a formula that looks roughly like this, where the probability is related to a constant that's going to capture concentrations and affinities, and it's going to apply both to cis and trans effects. And of course, it's how we think about trans effects that's proving the stumbling block. What we'd like to do is to run the formula on fibroblasts, rerun it after expressing in the computer the 4 Yamanaka factors at a higher concentration, and then compare the outcomes that we expect with the experimentally derived data from stem cells. And we would hope that the formula enables us to recapitulate what we see experimentally. And then we would like to go on to be able to predict which transcription factors you would need to overexpress to convert a stem cell into any cell type of your choice. So in this section, I've given you a simple formula that you can use to predict the activity of your favorite gene or enhancer. It contains just one variable, the number of base pairs to the nearest promoter, and it yields better results than the most widely used approach, RNA-seq. I hope you can see that the formula is easily extended by adding more Feynman diagrams. In the next section, I'll go on to wrap up the whole presentation. Thank you for your interest.